human heart, it's a muscle, but not like any other. It never rests. It beats 60 to 100 times a minute on average, all day, every day. Tens of thousands of times a day, tens of millions of times a year, a wave of electricity passes over it, making it contract, making it beat. If you live to be 80 years old, your heart will have beat about 3 billion times or more. How does it work? And what's happening when it stops? Welcome to Pocket Science, a compact guide to the human body and how it works, powered by the world-class scientists of the Fralin Biomedical Research Institute at VTC. Hey there, I'm your host, Matt Chittum, and today we're going to learn more about what makes the amazing heart do what it does from Steve Poelzing. Steve is a professor and longtime investigator here at the Research Institute and a member of the Institute's Center for Vascular and Heart Research. Thanks for joining us here on Pocket Science today, Steve.
Thanks, Matt, for having me.
So before we get to the science, you're a fit, active guy. What gets your heart rate up? I understand you're a rather fierce pickleball player.
I enjoy playing pickleball. I also very much enjoy mountain biking, particularly with my two boys. They really like going out on the trails. It's a great place in Roanoke, Virginia to get on the trails and think, oh boy, I hope I don't die today, and just ride.
So, hey, back to our big question. So what does make the heart beat? Is it really electrical, like the way we think of electricity outside the body? And how is that electricity conducted across the heart? I understand there's not necessarily complete agreement out there on how this works. So answer that question for us first.
Okay, so the electrical activity is made actually by salts being on inside and outside of the heart cells. There's really three salts you have to know about. The first is sodium. Think of sodium like the super fan outside of a stadium. There are lots and lots of them, and they bring the energy from outside of the heart cell into it. The second player is calcium. Calcium are the athletes. They sit inside of a locker room largely, and when sodium comes in and starts up the heart cell, calcium comes rushing out. It binds to the contractile machinery and kind of like causes all the action to occur. And then finally, there's potassium. Potassium is the salt that is, its job is, they're kind of like the everything. They clean it up. They clean the stadium. They open the doors. They close the doors. They just kind of reset. So their job is to go out of the cell and kind of pull all of that sodium back out with them and reset the heartbeat. So to your follow-up question of, well, how does it get from one cell to the other? Most scientists think that there are a protein called a gap junction. You can think of them like sky bridges between stadiums, for my analogy. And these sky bridges allow, when the first heart cell fills up with sodium and a lot of activity happens, it pushes all of that potassium into the next cell, which starts opening doors on the next cell. That causes sodium to rush into that next cell and calcium to start contraction. And then it just repeats over and over and over until all the heart cells are synchronized in order every single time. So that's the predominant theory. Do you want me to go into the controversy?
Yeah. So what's the other idea out here? And I think you subscribe to this alternate theory, I guess, or it's not an alternate, it's yes and, I guess.
It is a yes and. In the 1950s, they proposed that there could be gap junctional coupling between heart cells. They had ruled out that heart cells communicate like neurons, so heart cells don't really communicate with each other through synapses. Instead, when the heart cells get really, really close together, it's actually the loss of sodium outside of the heart cells where they're joined that actually causes the doors on the other cell to open. So the easiest way to imagine this is like static electricity. That all of that movement of sodium into one cell across that little gap, that very large flux causes the next cell to just kind of spark open, and then the sodium changes direction and runs into the next cell. And that happens faster than you can get salts to cross those gap junction sky bridges.
So the gap junction is more like a coupling, and then the ephaptic coupling more like jumps the gap by itself, like the way static electricity jumps from a shirt when I get ready to pick it up out of the dryer.
That's exactly it.
Wow.
Yeah.
And am I right that it moves across the heart like a wave, cell to cell, and that causes them to contract in the right order, ideally, and that's the heartbeat?
That is. Yep. It starts in a tiny little group of cells called the sinoatrial node. It's right at the top of your heart. Those cells set their own rhythm by their own mechanism. They then push out that first beat, and then it goes into the atria, which are the top of the cells, down into the big muscles, which are the ventricles, which squish the blood into both the lungs and the body. And every single cell. So for your beautiful math on the billions of heartbeats, there are billions of cells that are experiencing perfectly synchronized billions of heartbeats every single lifetime.
Okay, just stop right there. You're blowing my mind.
Okay. You did the math. I enjoyed the math.
Hey, sorry to interrupt, but Steve really stopped me with that observation. The sheer number of times your heart beats in a day or a year or a lifetime is incredible. But when you think about what's involved in just a single heartbeat, it's even more stunning. For the heart to beat just once, electricity has to pass from one cell to the next across all 2 or 3 billion heart muscle cells in just the right timing and just the right sequence. We're talking about a perfect symphony played at least once a second for as long as you're alive. That it works even most of the time is incredible. That it happens every time? Yeah, mind blown. So it's an amazing process, but it doesn't always work, right? And when it doesn't, it can be even fatal. And if I have this right, you don't often see it coming. Your lab focuses on problems with this electrical conduction process and how it can lead to sudden cardiac death, in the worst case scenario, I guess. So a lot of these problems are hard to identify and harder to predict, right? So talk about what your lab is learning and where that might lead.
The electrical, if you imagine the electrical wavefront truly as a wave, so imagine like a river with a wave going down it. If a piece of tissue dies, for example, in this analogy, there's a rock in the middle of that electrical wavefront. If you look at a wave that hits a rock, on the backside of it, you'll see little eddy currents form. They're little spirals. So you can imagine that if the heart, which is supposed to have this beautiful wave that moves across it every single time, if it starts developing these little tiny eddy currents, those eddy currents spiral off, and then the heart starts quivering, which is called fibrillation, instead of contracting in its synchronized activity. But unlike the river analogy, once that little spiral wave sets up in the heart, it's very hard to terminate. And this is why there are defibrillators out there. But as we go to your first point of, you can't predict it. To date, no one knows when you're going to have an arrhythmia. We have risk factors. Some of them are genetic. So if you're born with a mutation on a protein, or a family member has a mutation on a protein and they die suddenly, you'll be whisked into the doctor's office to be genotyped, and you'll ask inevitably, when's my number up? And just because you have a mutation doesn't mean you're going to manifest it. They'll run all kinds of tests on you. So what do? We found that there is a class of mutations. These are all mutations in those tiny little spaces between the heart cells, so next to the gap junctions and even the gap junctions themselves.
Okay, let's talk a little more about those tiny spaces between heart cells. How tiny are they? We're talking about a gap of between less than 30 nanometers up to about 100 nanometers. So what does that mean? American paper money is about 100,000 nanometers thick. So that means Steve and other cardiovascular scientists are peering into and identifying problems in a space equal to about 1/3000th of the thickness of a dollar bill. Okay, back to the interview.
That when they have mutations, or during our normal aging process, sometimes proteins in these little tiny spaces between the cells start to decrease their functionality. They get tired effectively. And we found that if you have both ephaptic coupling and gap junctional coupling, everything's good. It turns out that under a lot of diseases, we lose our gap junctional coupling pretty quickly. Gap junctions can remodel, or they can change their total expression in your heart in under 30 minutes. So they're really fast remodelers. And you can imagine over a lifetime that you can lose a substantial number of these proteins. But as long as you have some, we believe, as long as you have some ephaptic coupling to balance it out, the wavefront can still jump from cell to cell. Our research is showing that if you have the disruption of both gap junctions and ephaptic coupling, that's when the risk for sudden cardiac death becomes very predictable.
Got you. So once you've identified that, what can you do with that information, or what do you imagine could be done with that information?
Right now, we're discovering a number of ways that modify the spaces between the cells. So we're not actually focusing on the gap junctions. There are a number of individuals and scientists and great groups who are working on trying to prevent the loss of gap junctions. We're taking a slightly different view that nature has already evolved a way to account for those loss of gap junctions, and it's called ephaptic coupling. And so our research is really targeted on keeping that EFAPs narrow, or that little space between the cells very narrow. One way of doing that is making sure that your ions are properly balanced, that the space is regulated by the composition of the ions in that little cleft. And, if I projected in the future, one day I would hope that we would have a test that could tell you, are your ions in balance sufficiently to promote ephaptic coupling, and B, are these little spaces, these ephapses, are they appropriately close? Because I think a lot of individuals who have heart disease and have defibrillators, pacemakers, or there's a significant psychological burden to wondering, is today the day? And as you just did with some beautiful math, even if you're living, to your 20s, 30s, 40s, 50s, and 60s, you've had still billions of normal heartbeats. So the default of it's okay, your heart is okay, is more frequent than the arrhythmia. The arrhythmia, sadly, is a once-in-a-lifetime event. So it'd be nice to just give patients, I think, the understanding that today is going to be a good day. And if it's not, then they can have the peace of mind to go to the doctor and get their electrolytes corrected, get some therapy that is designed to actually push their heart back into the normal state.
Wow. Just ponder that for a minute. What Steve is describing is one day potentially being able to use a simple test to tell if these factors that can trigger a fatal heart rhythm problem are all good or in need of attention. If you have a heart problem that makes you more susceptible to an arrhythmia, it's like wondering if this little bomb inside you is going to go off and when. But what if you could start your day or your week knowing if that's more or less likely? And by knowing, you could maybe do something to prevent it. That's not just improved heart health Steve's envisioning. It's quality of life. It's like Steve said, it's peace of mind. So I listened to your TED Talk, which was, you were a somewhat younger man then.
I had hair.
This is not a visual product. So
I know.
You volunteered that. I didn't go there. And I've also heard you talk about and that has a lot to do with sodium. And you mentioned sodium earlier. That some of the therapeutic possibilities here may just be as simple as sodium. I mean, am I wrong about that? Tell me, straighten me out, or tell me what I'm thinking of.
Sodium is one of the most important salts in the entire body. So it's important for the heart. It's important for your kidneys. It's important for your digestive, your brain, everything. So sodium, as I said, it sits outside of the cells of all the cells in your body. And it triggers these electrical events all over. But specifically for the heart, maintaining the perfect balance of sodium outside the cell is it's a cooperative physiological event with your kidneys, as I said, your stomach, your intestinal tract. Like your body, even your lungs are working really hard to keep these salts in balance all the time.
Wow. And so could a therapy be as simple as adjusting the amount of sodium?
There are a number of diseases where already physicians will give a specific bag of high sodium salt water to patients. And we think that with some of our research that there are specific diseases where you can dial in the sodium to keep that disease in its concealed phase.
This is fantastic stuff, Steve. I'm learning every second here. It makes me wonder what gets you out of bed and making your instant coffee and coming into the office in the morning. What gets you into the lab and keeps you going?
I'm going to back up a little. My advisor when I was a PhD student was a cardiologist. He was a physician. So he was training us to really appreciate that what we do in the lab is not just reductionist science. At the other end of what we're doing, we always have to think of, is this going to help a person or groups of people? And so I guess what really gets me out of the lab is I think, oh, this is really exciting. No one else in the world has quite figured out what we figured out. And we show up in the lab and we test hypotheses all day. And then we think about, okay, is this, I guess for me, what's even more important is this a cost-effective and easy to implement solution to a very complicated problem? And that can be used as an adjuvant, like an additional therapy to some of the great medications, devices, and drugs we already have. But if you can very cheaply balance your salt water and fix a cardiac disease, that to me, I think would serve a lot of people very well.
I could see how being in the lab, you would, it does become, as you say, reductionist. It's about the task in front of you and did this work or not work and what's the next step. And you're not a physician. You don't have a stream of patients in front of you. What do you do to keep yourself mindful of those patients that might benefit your science one day down the road?
A couple of things. First, I do collaborate a lot with physicians. So a lot of I'd call them my scientific friends. At Carilion Hospital System, I collaborate with individuals in cardiology and cardiothoracic surgery. And it's very humbling to listen to them and ask them, what are your needs? What do you think are the unaddressed problems in your field? And then I share my ideas with them and they say, okay, I can see that might work. Let's try it out. So that's the direct connection. Otherwise, I'm fortunate I get contacted by physicians periodically and they ask me about a particular case and I remind them I'm not a physician and they say, yeah. Now tell us about your research. And then some of the things I've been most proud about are one-off case studies where I've worked with a group in Pennsylvania. They had a patient and they asked about a study we had conducted and not in humans. And at the end of it, they took a child who was having a lot of arrhythmias every single day and they did this intervention. I held my breath. I'm not as brave as an MD, but I held my breath and it reduced the child's arrhythmia burden. So I get a lot of opportunity to kind of see why this matters.
You're doing fantastic work, and it's really important work. I think if it were me, if I knew what you know about what can go wrong in the heart, it would keep me paranoid all day long. And if I, for example, was you and I stepped off the pickleball court, would I be going, what was that? What was that? Something felt funny. But you seem well adjusted, unlike maybe not everybody else in this room right now.
Well, let me see if I can change your frame then just a little bit. I like you, when I first started this, was very interested in wow, it seems like everything's going to fall apart instantly all the time. But the more I've been doing this, the more I realize that these aren't necessarily redundant processes in the body. There are a number of processes, each doing their own thing, that come together to make this emergent system that we call life. So even though we spend a lot of time talking about sodium, it's not just simply if you have high or low sodium. It can be adjusted for based on, well, what's the calcium that day and what's the potassium that day, or even some other proteins or other salts. So after studying this for a long time, I'm actually far less concerned about having everything perfect, because I do recognize that I've had many years on this planet of perfectly normal heartbeats, and I will continue to have a lot more perfectly normal heartbeats. So don't stress too much is my advice, because that one is a whole other field of research and not good for your heart or anything else.
Well, I'm not the smartest guy in this room right now, but I'm smart enough to know that that's the right place to stop. Steve, thanks for coming in. Again, Steve Poelzing. He's a professor here at the Fralin Biomedical Research Institute and a member of our Center for Vascular and Heart Research, and we're lucky to have him here.
Thanks, Matt.
That's it for this episode of Pocket Science. If you like what you heard, follow us and subscribe to Pocket Science on your favorite podcast platform. Or find us on the Fralin Biomedical Research Institute website, fbri.vt.edu. The Research Institute is a part of Virginia Tech. Pocket Science is produced and engineered by Clayton Metz, and I'm your host, Matt Chittum. Till we meet again, see ya.